Today's topic is translation. At the beginning, I will tell you about the genetic code because it is essential to know about the genetic code to understand translation. Now, a triplet sequence of the nucleotides A, U, G and C is the code word or codon for each amino acid. The complete set of these codons is the genetic code. Features of the genetic code. Number 1, triplet. As proteins contain the same 20 amino acids, there must be 20 separate codons. Since there are only 4 different nucleotides in mRNA, each codon must consist of more than one nucleotide. Codons consisting of 3 nucleotides can provide for 4 cube, that is 4 to the power 3, that is 64 codons. Thus, each codon is a sequence of 3 nucleotides on mRNA. For example, AUG codes for methionine. Number 2, universal. The same codons are assigned to the same amino acids in all living organisms. But there are few exceptions. For example, in mitochondria, AUA codes for methionine. Number 3, non-overlapping. Reading of genetic code does not overlap. Number 4, continuous. The codons are continuous, and once the reading starts at a codon, consecutive codons are read without punctuation or stopping, and stops when a terminator codon is reached. Number 5, degenerate. 3 codons do not code for any amino acid, and these are known as terminator or nonsense codons. They specify where protein synthesis stops. The remaining 61 codons code for 20 amino acids. So obviously, multiple codons must code for the same amino acid. That is, the genetic code is degenerate. For example, GUU, GUC, GUA and GUG. That is, the first 2 are GU and the third is UC, A and G. All these code for valine. Usually, the first 2 nucleotides rather than the third determine which amino acid will be coded. 6. Unambiguous. Any individual codon codes for only one amino acid. For example, GUU codes for valine only. Now, you must remember the difference between degenerate and unambiguous. For example, I just told you GUU and GUC both code for valine, but this is degenerate, and unambiguous means GUU codes for valine only, not for any other amino acid. So for valine there are many codons, but for one codon there is only one amino acid. Lastly, wobble. Base pairing between the third nucleotide of a codon and the corresponding nucleotide in the anticodon is not strict. That is, the coupling of codon and anticodon can be unsteady. For example, the four main wobble base pairs are guanine uracil, hypoxanthine uracil, hypoxanthine adenine, and hypoxanthine cytosine. So this is the genetic code. The first letter UCAG, the second letter UCAG. So here you can see for this one the first letter is U, the second letter is U, and the third letter is U here. For this one the first letter is U, the second letter is U, the third letter is C. So this becomes UUC. In this way you can see the tabular form of the genetic code. UUU, UUC, these two codons code for phenylalanine, and so on. You must remember AUG codes for methionine, and these three UAA, UAG and UGA are stop codons or nonsense codons. Now protein synthesis or translation proper. Protein synthesis or translation is divided into four parts: activation of amino acid, initiation, elongation and termination. Now the details of each part. First, activation of amino acid. Each tRNA is charged with its specific amino acid by an aminoacyl tRNA synthetase. For example, this tRNA will take tryptophan. This is tryptophan and this is the enzyme. So in the enzyme, tRNA synthetase is attached tryptophan, and the tRNA reaction occurs where ATP is broken down to AMP and the amino acid is linked to tRNA here by a high energy bond. So I'll repeat, each tRNA is charged with its specific amino acid by an aminoacyl tRNA synthetase, which attaches the carboxyl of the amino acid to the 3' hydroxyl adenosine terminal of the particular tRNA. So here we get an aminoacyl tRNA. Next, initiation. Initiation is divided into number 1, ribosome splits into 40S and 60S subunits. Number 2, the 40S combines with methionine tRNA GTP eukaryotic initiation factor 2 forming, that is 40S plus met tRNA GTP and EIF2 together form pre-initiation complex. This 40S pre-initiation complex along with mRNA forms 40S initiation complex. Lastly, 40S initiation complex combines with 60S subunit to form 80S initiation complex. Now the details of each step. Number 1, eukaryotic initiation factor 3 and eukaryotic initiation factor 1A bind to 40S. This helps ATS to split into 40S and 60S subunits. Number 2, GTP binds to EIF2, that is eukaryotic initiation factor 2. These then bind to MET tRNA. Then all these bind to 40S subunit and form pre-initiation complex, which is stabilized by EIF3 and EIF1A. EIF2 has alpha and beta subunits. EIF2 alpha is phosphorylated by a cyclic MP independent kinase, which is activated when cell is stressed, as in viral infections, heat shock, low amino acid or glucose levels. EIF2 alpha inactivates 2 beta, leading to block of GTP GDP recycling, resulting in stoppage of translation. Number 3, 40S pre-initiation complex is bound by mRNA cap. These together then scan mRNA for AUG codon near a Kozak consensus sequence. EIF4F complex, which controls rate of translation, consists of EIF4E, EIF4A and P220. Overexpression of EIF4E causes malignancy and underexpression leads to prolonged cell cycle. Insulin activates EIF4E, so there is more protein synthesis in muscle and adipose tissue. Number 4, EIF5 dissociates GTP from EIF2, causing release of initiation factors bound to 40S initiation complex. Thus, free 40S binds to 60S subunit, forming ATS. The initiation factors are recycled. Next, elongation. Elongation includes binding of amino-acyl tRNA to A site, then peptide bond formation, lastly translocation. Now, details of each part. Number 1, EEF1 alpha, that is eukaryotic elongation factor 1 alpha, complexes with GTP and proper amino-acyl tRNA. The tRNA then enters the empty A site. This is the A site. This is a tRNA with 1 amino acid. So this is amino-acyl tRNA. This enters the empty A site. Also, EEF1 alpha GDP is released. EEF1 alpha GDP is recycled to EEF1 alpha GTP. So this was the binding of amino acid tRNA to A site. Next, peptide bond formation. Amino group of amino acid tRNA in A site bonds with carboxyl group of peptidyl tRNA in P site catalyzed by peptidyl transferase of 28S rRNA. The whole peptide is transferred from tRNA in P site to T site I'll repeat. Then whole peptide is transferred from tRNA in P site to tRNA in A site. So from P site to A site. From P site to A site. Number 3, translocation. The tRNA in P site is removed. EEF2 and GTP transfer the tRNA with peptide from A site to P site. So from A site to P site. The whole cycle is repeated. Lastly, termination. When a terminator codon appears in the A site, ERF, that is eukaryotic release factor, GTP and peptidyl transferase hydrolyzes the bond between peptide and tRNA in P site. Now, after translation, we have two more little topics, that is post-translational modifications and inhibitors of translation. Modifications occur after protein synthesis, that is after translation. These are called post-translational modifications. For example, important ones like number 1, gamma carboxylation of glutamic acid in prothrombin by vitamin K. This is important, and I think you'll be taught this in vitamin K. Next, hydroxylation of proline and lysine in collagen by vitamin C. This also will be taught in vitamin C. Then disulfide bond formation in insulin, that is intrachain and interchain bonds. If you have studied the structure of insulin, you know that there are bonds between the two chains of insulin and also within each chain of insulin. There are many other post-translational modifications which are essential for making the protein active. Inhibitors of translation. All these are inhibitors in prokaryotes. This year you had a question, MCQ, inhibitor of translation in eukaryote. You had the choices streptomycin, erythromycin, chloramphenicol and puromycin. Anyway, bacteriostatic, that is inhibitors which cause stoppage of growth of a bacteria. They don't kill the bacteria as such. And bactericidal, that is inhibitors which kill the bacteria. So bacteriostatic, like tetracyclines. You also need to know how the inhibitors act. Tetracyclines inhibit attachment of amino acyl tRNA to A site. Next, chloramphenicol inhibits peptidyl transferase. Erythromycin prevents translocation. Next, bactericidal. Streptomycin causes misreading of mRNA and inhibits initiation. I also might mention diphtheria toxin attaches ADP to elongation factor 2 and therefore inactivates elongation factor 2. So this is all for today. Thank you.